This is lab 11. In this lab, you're going to grow bacteria from the environment and perform two different biochemical tests. So there are going to be two labs, biochemical test 1 and 2. But the first thing you're going to have to do is grow bacteria. Before you start the lab, like all the other ones, make sure you disinfect your surfaces, wash your hands, and wear all the appropriate PPE. Experiment, so lab 11, the beginning of biochemical testing 1. Inoculation of plates, and so you're going to need 2 nutrient agar plates that I've already poured, and so they're ready to go. You're going to use a Sharpie to, on the bottom of the plates, not the lid, divide them in half. So I've just marked each of the halves, and they're going to be then 4 samples that you have. So the recommendations is a sink, kitchen sink works well, and then something like a shoe, a phone, and then we like we normally do, we'll include a control. So we're going to be sampling with our sterile swabs. The packages that I have 2 swabs each, so you'll need 2 packages or 4 swabs each. And then I have some distilled water. As we've done before, it's helpful to moisten the sterile swab with some distilled water so that it gets a little better sample of the bacteria. So I'm going to do the shoe first, and then I'll do the other ones as well. So moisten the sterile swab, and then just get a good sample. So this is the shoe side. So again, whenever we do this, we try to hold the lid over so it helps prevent the airborne contaminants from falling onto the plate. And next is the phone. So I'm recording with the phone right now, so I'm sampling from it. So this is the phone side. I'm going to do the control, so this will be done the same way, except I'm not going to, so I did dip it in the water, but then and the last one is the sink. So I'm just going to get down in there, roll it around. So my sink is this one here. So as we've done in previous labs, we're going to parafilm these and then put them to incubate, and we'll be doing something with the bacterial colonies that arise after the incubation. So this is a continuation of biochemical testing number 1. And so this is specifically how to perform the oxidase test. And so this oxidase cassette, you should have been keeping it in your fridge. It was one of the items that upon receipt of your kit, it indicated to store it in the refrigerator. So now we are going to use it. The other thing you're going to need is some isopropyl alcohol to sterilize your inoculating loop. And so I always use a graduated cylinder for that. So I have that filled. And then you'll need your tea light and your matches to light it, and then of course your loop. So we grew bacteria in different conditions. So I have my sink and shoe plate here. My sink grew really crazy. I have a bunch of bacteria. My shoe just has some other things on it. My other plate, my control, and my phone didn't grow extremely well. So ideally, what you're going to do is use 3 of these areas from bacteria from 3 different areas sampled. So 1 sink, 1 shoe, and then some local surface. If you don't have growth on a plate on a specific region like I have, I don't have anything really growing on my phone, then just take duplicate samples from shoe and sink. So just choose a different bacteria. So based on what the colony looks like, just sample a different area. So I'm going to sterilize my loop. So remember, about 10 seconds submerged in isopropyl alcohol. And I use the graduated cylinder so I can get the entire wire area submerged in the isopropyl alcohol. And then once that 10 seconds is up, you just burn off that alcohol. And most importantly, make sure that loop area glows kind of red hot so that you know you're up to temperature that will incinerate any living biological material. And then of course you need to let it cool. So in this first zone, I'm going to take a sample of my sink bacteria. So this is now cool. I'm gonna get some of that bacteria, and then you just put it. And what you're looking for is a color change to a blue or purple in 30 seconds or less. And so this is already turning purple. So I'm gonna choose another sink bacteria so you can see the color starting to develop there. So that would be a positive result. And what that tells you is this bacteria that I just put in that top left square does indeed have the enzyme oxidase. So it's short for cytochrome oxidase. So the oxidase test is asking the question, does this bacteria have the enzyme oxidase? And so a purple result in 30 seconds or under means yes, the bacteria does have the enzyme oxidase. And then hopefully I'll have one that's negative to show you. So I'll do a shoe bacteria now. So this is now, and you want to get a good amount of bacteria. So that's the second bacteria there from my shoe. And that doesn't look like it's changing. So luckily I got one positive result and one negative result to show you. I'll do another sink colony because likely that will be positive as well in this third area, and we'll see what happens. Trying to spread it out more so you can see it better. So the color's already starting to develop on that one. So again, it has to turn a dark blue or purple in 30 seconds or under for a positive result. Okay, and so what we have here is a positive result, a negative result, and another positive from a different bacterial colony from my sink sample. But that's what you're looking for with an oxidase test. And this is called a biochemical test because we're testing for the presence or absence of a specific enzyme, which is part of the biochemical makeup of a particular organism. So enzymes are really important in biochemical or metabolic pathways. And so we can ask questions about specific enzymes. Are they present or not? And the way that we do that is we look for the ability of that enzyme to carry out a certain reaction, either in a tube or in this case, kind of on a little cassette. So again, this bacteria positive in the oxidase test means it has the oxidase enzyme. This one also was positive, so it has the oxidase enzyme. And this one didn't turn purple within 30 seconds, so it's negative. It does not have the oxidase enzyme. After you've done the oxidase test on the dry slide cassette, make sure you take a photo of it. That's going to be one thing that you submit with your homework assignment. And then once you have that photo, you can discard the dry slide cassette, but don't discard your bacteria because you need them to perform part 2 of this biochemical test experiment. So save those bacteria, you're going to use them in the next part of the video. So this is a continuation of the biochemical test part 2. And we're going to use the same bacteria that we used in part 1 for the oxidase test, but now we're going to do a test that's called the catalase test. And so based on that name, catalase is also an enzyme. So the catalase test is specifically asking the question, does a particular bacterial species have the enzyme catalase, or does it not have the enzyme catalase? So does it have it or does it lack it? And so to do this test, you're going to need an empty Petri dish. And what I've done is I've divided it into thirds. And these thirds are going to represent the different locations that the bacteria were sampled from. So you should have shoe, sink, and local. So again, my phone doesn't have a good growth on it. And so what I'm going to do is just do 2 from shoe, 2 from sink, and then I'll choose another couple colonies just to get 6 bacterial colonies tested completely. But I'm going to start with the sink. And the way that we're going to do this is, again, using our loop. We sterilize it in our isopropyl alcohol. So it needs to be submerged for about 10 seconds. And then once it comes out, you can flame it, burning off all the isopropyl alcohol, but then also making sure that that loop portion is red hot. And then we'll have to let it cool. So I'm going to do the sink first. So flip that over. So I'm going to do these areas. I'm going to grab a colony. And this plate doesn't have any agar in it. It's just, I'm going to smear the bacteria in a circle and then burn this off. And what I'm going to need is, from your kit in the bag that has the tubes of different reagents in it, you're going to find a small tube of hydrogen peroxide. And we're going to use that in just a second. So using the pipette pump and a sterile pipette, we're going to draw up this hydrogen peroxide. And then what I'm going to do is I'm going to put 1 to 2 drops in that circle. So I'm going to not touch the bacteria, but I'm just going to drop it directly on it. And what I'm looking for is the presence of bubbles. So if the bacteria has the enzyme catalase, then this hydrogen peroxide is going to serve as the substrate, or that's the fancy word for the enzyme is going to interact with this hydrogen peroxide and convert it to water and oxygen. And so that oxygen is a gas, and so you should see bubbles. So I have no idea how this one's going to be, but I'm just going to drop, oops, I missed 2 drops. And I don't know if you'll be able to see that well, but there are bubbles that are forming there. And so that would be a positive catalase test. So those bubbles that are forming would be positive for catalase test. So now what I'm going to do is just sample another bacterial colony. And I'll do a shoe this time. So sterilize your loop in the isopropyl alcohol. Burn off all of that isopropyl alcohol and then get the loop part red hot. And then we will, of course, let it cool. So I'm going to do this shoe zone next. So I'm going to get a colony. For my shoe. And then sterilize your loop. So I still have hydrogen peroxide in this one, so I'm going to drop 2 drops. So this one too is positive. You can see bubbles forming pretty quickly on it. So that shoe colony also is catalase positive. What we're looking for a negative result would be a bacterial colony that doesn't yield any bubbles. So if you get a good amount of the colony and it doesn't yield any bubbles, then that would be a sign that the bacteria doesn't have the enzyme catalase. So far, the two that I've chosen are catalase positive because we do see bubbles. So I'll do another one with the hopes that it will be negative. So I'm going to do another shoe colony. So I'm going to drop 2 drops of the peroxide. So that one doesn't seem to have it. You can see the other ones are still have bubbles forming, indicating they're catalase positive.
After you're done performing the catalase test, make sure you take a photo of this dish that has the different catalase results in it. It, along with the dry slide picture you will submit with your assignment. Once you've done that, you're done with all the plates from this experiment. So the catalase plate you can bleach, and then the 2 nutrient auger plates that have the bacteria that can be bleached as well. So remember, that's 20 minutes with full strength bleach, and then it can be poured down the drain. And then parafilm the plates before they're put into the garbage. And then because we're done, you can disinfect the surfaces and wash your hands.